I'm 71 years old now, and for 34 years I've been carrying knowledge that contradicts everything we think we know about where they come from. My name is Dr. Marcus Feldman. I'm a retired molecular biologist, and in 1990 I was given access to biological material that changed my understanding of reality forever. I spent 30 years at MIT focusing on comparative genomics and evolutionary biology. My career was unremarkable in the best way, publishing papers, teaching graduate students, running a small lab funded by the usual grants. I lived in Cambridge with my wife Ellen, who taught literature at Boston University. We had 2 daughters, a mortgage, and a life that felt satisfyingly ordinary. In March of 1990, I received a phone call from Dr. Robert Kerrigan from an organization called the Advanced Biological Research Division. He said they were assembling a team of geneticists for a classified government contract, and that my work on comparative genome analysis had caught their attention. The pay was exceptional, nearly triple my university salary for an 18-month contract. The catch was that I couldn't discuss the nature of the work with anyone, including my family. The security clearance process was invasive. They interviewed colleagues, neighbors, reviewed my financial records, my travel history, everything. In May of 1990, I received notification that I'd been cleared for a project designated only by a number. I told Ellen I'd been selected for classified defense research, which was technically accurate. I promised I'd be home every few weeks. I flew into Albuquerque on a Monday afternoon in early June. A driver met me at the airport and we drove for nearly 2 hours into increasingly remote terrain. The facility looked like a small industrial complex from the outside, nothing suggesting what actually happened inside those walls. The security was unlike anything I'd experienced in civilian research. Biometric scanning, multiple checkpoints, surrender of all personal electronics. A woman in a lab coat, Dr. Sarah Chen, escorted me through corridors that descended gradually underground. She explained that I'd be working primarily on level 3, conducting comprehensive biological analysis of specimens recovered under classified circumstances. The specimens were of unknown origin, and our task was to determine everything we could about their biology and genetics. A military officer added one final detail. Everything we discovered was classified at the highest level. We would work in compartmentalized teams, and violation of security protocols would result in immediate prosecution. Then he said something that made my stomach tighten. He said, "Whatever conclusions you reach, document everything, but remember that some truths are more complicated than they first appear." On June 5, 1990, they took me down to level 4 and showed me what I'd actually be analyzing. That was the day my understanding of biology shattered into pieces. I'm still trying to reassemble. The examination room maintained a constant 62 degrees. Dr. Chen walked beside me, explaining that the specimen had been kept in specialized preservation since its recovery using classified methods. The specimen was in the center of the room, on a tilted examination table under bright surgical lights. It was small, perhaps 4 feet tall, with a body structure that was humanoid but wrong in its proportions. The head was disproportionately large, maybe a third of the total body mass, with a cranium that extended back and up in a way human skulls don't. The skin had a gray-green tone with a texture that appeared smooth but somehow dense. The eyes were enormous black ovals without visible pupils. The nose was barely present, just 2 small slits. The mouth was a thin line, almost lipless. The limbs were elongated, particularly the arms. Each hand had 4 digits, longer and more slender than human fingers. I asked where this came from. Dr. Chen said that information was compartmentalized above my clearance level, but added that it had been in government possession since 1947. That date sent a clear message, even if she couldn't speak it directly. The preservation was remarkable, unlike any taxidermy or medical preservation I'd encountered. The tissue appeared supple, maintained at a cellular level that shouldn't have been possible for something supposedly 43 years old. My task was to conduct comprehensive genetic analysis, determine cellular structure, compare it to known terrestrial organisms, and document any anomalies. I spent the first day documenting external features, taking measurements, photographing from multiple angles. The body was symmetrical in most respects, suggesting evolutionary pressures similar to those that shaped bilateral symmetry in Earth life. That detail troubled me more than the differences. Under the microscope, the cells appeared organized in recognizable patterns, membranes and nuclei, and organelles arranged in ways that made sense. But the more I examined them, the more I noticed subtle irregularities. The cell walls had different density than mammalian cells. The mitochondria had an unusual configuration I'd never seen in any terrestrial organism. I spent a week on cellular analysis before moving to genetic material extraction. Standard extraction protocols didn't work efficiently on this tissue. The cellular bonds were stronger, requiring different chemical treatments. I had to modify our procedures, testing different enzymatic combinations over several weeks. By the end of June, I had successfully isolated what appeared to be DNA. The structure looked right under electron microscopy, that familiar double helix. Seeing that familiar structure should have been reassuring. Instead, it raised more questions. If this was truly extraterrestrial, why would it use the same genetic storage system that evolved on Earth? I began sequencing on July 2nd. The data that came back from those first runs made me check the equipment calibration 3 times. The genetic code was readable. It used the same 4 nucleotide bases as terrestrial DNA, but the sequences themselves contained patterns I'd never encountered. Some sections looked almost familiar, like distorted versions of genes I recognized. Other sections were completely alien. I worked alone in that laboratory for 14 hours a day. Dr. Chen would check on my progress weekly, but offered no insights. The other scientists were working on different aspects, different specimens, but we never compared notes. The isolation made the work feel surreal. By late July, I ran the sequences through our database, looking for matches to any cataloged organism. What came back made me sit in silence for a long time. The results suggested something that violated everything I thought I knew about biology. The comparative analysis results appeared on my screen at 11:47 p.m. on July 26, 1990. I had run the comparison 3 times over that week, convinced a species was seeing contamination, but the results remained consistent. Sections of the specimen's genome matched terrestrial DNA with precision that couldn't be coincidental. The matches weren't random fragments. They were functional gene sequences. I found a sequence 87% identical to a human gene involved in neural development. Another section matched primate DNA at 76% similarity, specifically genes associated with cognitive function. There were matches to mammalian immune system genes, to metabolic pathway regulators, even to certain plant genes. Approximately 30% of the specimen's genome had clear terrestrial origins. The remaining 70% was unlike anything in our databases. It was as if someone had taken Earth biology and woven it together with something completely foreign. I tested the contamination hypothesis thoroughly, but the integration I was seeing wasn't surface contamination. These sequences were embedded within the genome's structure, flanked by the unknown genetic material in ways that suggested intentional organization. They were part of the original design. By early August, I had to face an impossible conclusion. The genetic material in this specimen didn't just resemble Earth biology. It contained actual Earth DNA, integrated at a fundamental level. Someone or something had deliberately combined terrestrial genetic material with whatever the other 70% represented. This wasn't natural evolution. This was engineering. I requested a meeting with Dr. Kerrigan on August 9th. He read through my findings in silence. When he finished, he asked if this could be explained by environmental exposure after arrival. I told him no, that environmental exposure doesn't rewrite an organism's genome. He told me to continue my analysis, but keep my conclusions tentative. Throughout the genome, I found marker sequences, short, repetitive patterns that didn't seem to code for anything directly. They occurred at regular intervals, like punctuation marks. I'd seen similar structures in artificially modified organisms, in laboratory bacteria engineered to carry specific genes. Researchers insert marker sequences to identify modified sections. Finding them here suggested intentional genetic engineering. In October, Dr. Chen brought tissue samples from a different specimen, recovered in 1964. I spent the next month analyzing its genetic material. The genome showed the same hybrid structure, the same integration of terrestrial and unknown DNA. But when I compared the two specimens directly, I found they were genetically similar but not identical. They had variations that suggested they weren't clones. They were related, like siblings or cousins. That finding had profound implications. If multiple specimens existed with genetic variation, that suggested a breeding population. And if they were being recovered periodically over decades, it meant they were continuing to arrive or operate on Earth. I began analyzing which Earth organisms had contributed genetic material. The primate DNA was from the hominid line, closely related to modern humans. But there were also plant genes, specifically sequences from organisms that existed only in certain geographic regions. I found matches to deep-sea algae, to a fungus that grows exclusively in temperate rainforests, to bacteria from the American Southwest. The geographic diversity suggested extensive sampling across Earth's biosphere. By December 1990, I had developed a working hypothesis. The specimens appeared to be artificially constructed biological entities, created by combining terrestrial genetic material with non-terrestrial components. The terrestrial DNA provided proven genetic solutions for specific biological needs. The result was a hybrid organism, neither fully of Earth nor fully alien. But that hypothesis required accepting disturbing premises. Someone had been collecting genetic samples from Earth, possibly for decades. Whoever created these beings had technology far beyond human capabilities in the 1940s and 60s. And the crashes might not be accidents. They might be something intentional. In January 1991, Dr. Kerrigan informed me they wanted to extend my participation. They had conducted similar analyses at other facilities, and they wanted me to review those findings. I agreed, though it meant another 6 months of lying to Ellen about my work. In February, they granted me access to a secure database containing analysis reports from 4 other research teams. 11 specimens total, recovered over a 39-year period from 1947 to 1986. Each had undergone varying levels of analysis, depending on the technology available. I spent 3 weeks reading through those reports. The consistency was striking. Every specimen showed the same hybrid genetic structure, the same integration of terrestrial and non-terrestrial DNA. The proportions varied slightly, but the overall pattern held. They were all constructed using the same basics, template. All specimens stood between 3.5 and 4.5 feet tall. All had the enlarged cranium, the large black eyes, the elongated limbs. This wasn't multiple different species. This was one design type, showing up repeatedly over decades. I created a timeline plotting each recovery event, the first in 1947, then nothing until 1952, another in 1957, then 3 in the 1960s, 2 in the 1970s, 2 more in the 1980s. The frequency seemed almost random, scattered across decades. But there was one detail in every recovery report. Every specimen had been recovered shortly after detection of unusual aerial phenomena in the area. Lights in the sky, objects moving in ways that defied conventional aircraft, radar anomalies. Strange sightings preceded every recovery by hours or days. The tissue preservation method troubled me. The protocol dated to 1948, just 1 year after the first recovery. Someone had developed sophisticated biological preservation methods specifically for these specimens within months of the first one being obtained. Either they had anticipated the need or they had prior experience. One afternoon in May, I mentioned my confusion about the purpose of these beings to Dr. Chen. She asked me a question that shifted my perspective. She said, "What if they're not meant to survive in space? What if they're designed specifically for Earth's environment? If these beings incorporated Earth DNA because they were designed for Earth, that changed everything. They weren't visitors who happened to crash here. They were created specifically to operate in Earth's environment. The crashes weren't failed interstellar journeys. They were something else. Perhaps delivery systems. Perhaps experiments that went wrong." By June 1991, my extended contract was ending. I declined to continue. I needed to return to my family, to work I could discuss with colleagues. Dr. Kerrigan reminded me of my obligations under the security protocols. I assured him Seymour understood. On June 28th, I flew back to Boston. I returned to MIT, resumed teaching, tried to slip back into academic life. But something had changed. I would sit in faculty meetings, and part of my mind would be back in that underground laboratory, staring at genetic sequences that shouldn't exist. I tried to resume normal research after 1991, but the work felt hollow. The questions I couldn't answer had burrowed into my mind. By 1993, I'd found a way to address some of those questions indirectly. I couldn't discuss the classified specimens, but I could research publicly available information about government biological research programs from the 1940s and 50s. The university library had extensive archives of declassified government documents. I spent afternoons reading through obscure reports about Cold War-era research programs. Buried in less redacted documents, I found hints of something more sophisticated than the public record suggested. Project Sunshine was one discovery, officially studying radiation absorption, but some technical appendices mentioned tissue sample collection from multiple sources, genetic analysis techniques that seemed advanced for the early 1950s. The scope suggested capabilities beyond what I thought existed at that time. I wasn't looking for conspiracies. I was looking for technological capability. If the specimens had been created using advanced genetic engineering, that engineering had to have happened somewhere, using equipment and knowledge that existed at the time. In 1995, I attended a conference in Washington, D.C. I met Dr. Harold Zimmerman, a retired researcher who had worked at Oak Ridge National Laboratory in the 1950s. Over drinks, I asked about the state of genetic analysis during his time there. He told me they were more advanced than most people realized, that military funding had accelerated certain techniques beyond what academic laboratories could achieve. Could they sequence DNA in the 1950s? Not comprehensively, he said, but they could identify and isolate specific genes, could perform crude analysis, could work with cellular cultures in ways that wouldn't be published for another decade. The techniques were rudimentary by modern standards, but they existed. They were just classified. That conversation confirmed what I'd begun to suspect. Human biological research in the classified world had been more sophisticated than public science reflected. The gap between classified and public knowledge was larger than I'd assumed. I began looking at the genetic markers with new perspective. The terrestrial DNA wasn't random. Specific genes had been selected for specific functions. Someone had curated Earth's genetic library, selecting optimal solutions for particular biological needs. In 1997, I found a declassified document from 1962 mentioning a biological survey program conducted by the Department of Defense. The technical scope included genetic characterization of extremophile organisms and cross-species gene expression analysis. The program had operated from 1958 through 1965, with sampling efforts in multiple countries and environments. The timing was notable. The specimens recovered in the 1960s would have been created during or shortly before that decade. A government program systematically collecting and analyzing genetic material during that same period seemed more than coincidental. By 1998, I'd compiled a personal archive of over 2,000 pages of declassified documents, scientific papers, and my own analysis. I couldn't share it with anyone. But for me, the pattern was clear. Someone had been systematically sampling Earth's biosphere, analyzing genetic material from diverse organisms, and incorporating that material into engineered biological entities. The crashes took on new meaning. If the beings weren't visitors from distant worlds but products created using Earth biology, their presence on Earth made more sense. They weren't traveling from somewhere else. They were designed for here. The crashes might represent failed deployments or transport accidents. What disturbed me most was the continuity. Recoveries spanning from 1947 to at least 1986 suggested an ongoing program. This wasn't a single incident. It was a sustained effort, operating over decades, producing multiple individuals of the same designed species. In late 1999, I received a call from Dr. Raymond Kellerman from Lawrence Livermore National Laboratory. He wanted to discuss my research into 1950s biological capabilities. We met in Chicago in February 2000. Kellerman had worked on classified biological research since the 1980s and had become interested in the historical development of the field. I shared some of what I'd found, carefully avoiding any reference to my classified work. I discussed the biological survey programs, the advanced capabilities that seemed to exist earlier than acknowledged, the compartmented projects that employed geneticists for unspecified research. Then Kellerman said something that made me realize I wasn't the only person asking these questions. He said, "You've been wondering if we were capable of creating hybrid organisms back then, haven't you?" I admitted I'd been considering that possibility. Kellerman nodded slowly. Then he told me something that would reshape my understanding of what I'd discovered. He said, "We were capable of it. But that doesn't mean we did it." Kellerman's statement hung in the air between us. "We were capable of it, but that doesn't mean we did it." I asked him to clarify what he meant. He looked around the coffee shop, confirming no one was within earshot, then leaned forward slightly. He said that by the late 1950s, certain classified programs had achieved genetic manipulation capabilities that wouldn't appear in open literature until the 1980s. They could isolate genes, transfer genetic material between organisms, create hybrid cells in laboratory conditions. The techniques were crude and the success rate was low, but the fundamental capability existed. However, he said, creating a viable multicellular organism with integrated genes from multiple species, an organism that could grow and function independently, that was exponentially more complex. I asked if it was impossible. He said no, not impossible, but it would have required infrastructure and resources beyond what any single program likely possessed. It would have needed extensive trial and error, decades of research, facilities capable of supporting long-term biological development. The scale would have been enormous. Then he asked me a question. He said, "Have you considered that the engineering might not have been done here? That the sampling of Earth biology could have happened elsewhere, by someone with more advanced capabilities?" The implication was clear. If non-human intelligence wanted to create beings compatible with Earth's environment, they would need Earth's genetic material. They could collect samples, transport them elsewhere, conduct the engineering in their own facilities with their own technology, then deploy the results back to Earth. The crashes we recovered weren't arrivals from distant solar systems. They were returns to the source of their biological components. I sat back, processing this perspective. It explained the terrestrial DNA integration without requiring impossible human capabilities in the 1940s. It explained why the beings seemed designed for Earth rather than adapted to it. It explained the periodic recoveries, if these were ongoing deployments or experiments rather than random accidents. Kellerman told me he'd reached similar conclusions, working on a different classified project in the 1980s. He wouldn't specify what project, but he'd seen biological material that showed the same hybrid characteristics, the same integration of terrestrial and non-terrestrial elements. His team had concluded that the engineering was beyond human capability at the time of creation, which meant either humans had help or humans weren't the engineers. We talked for another hour, carefully dancing around the classified details we couldn't share while building a composite picture from what we could discuss. When we parted, Kellerman gave me his secure email address and said if I ever learned anything new, he'd be interested in comparing notes. I realized then that there were others like us, scientists who had glimpsed pieces of this puzzle through classified work, all of us bound by secrecy, but quietly trying to understand the larger truth. Between 2000 and 2005, I continued researching while maintaining my academic career at MIT. I focused on the history of genetic science, publishing papers on the development of DNA analysis techniques and the gap between classified and public research capabilities. The papers were well received in academic circles, seen as interesting historical work with no particularly controversial implications. But my private research went deeper. I began investigating the infrastructure that would have been required to support the kind of biological engineering I'd witnessed. Creating hybrid organisms wasn't just about genetic manipulation. It required facilities to grow and maintain specimens, equipment to monitor their development, expertise across multiple disciplines, from genetics to developmental biology to biochemistry. I found references to several large biological research facilities constructed in the Southwest during the late 1940s and early 1950s. Officially, these were studying radiation effects on living organisms, developing medical countermeasures for nuclear warfare, researching biological weapons defense. The facilities employed hundreds of scientists and technicians, consumed enormous budgets, and operated under strict security protocols. What interested me was the timing and location. Several of these facilities were constructed within a year of the 1947 recovery event. They were located in remote areas of Nevada, New Mexico, and Arizona, the same general region where most of the recovery events had occurred. The official justifications for their locations cited isolation for dangerous biological work, but the geographic clustering seemed significant. I also discovered that several prominent geneticists had disappeared from academic literature during the early 1950s. They would publish regularly, then suddenly stop, sometimes for years. When they reappeared in the public record, their research had often shifted focus entirely. I tracked down a few of these scientists, now elderly or deceased, and in the cases where I could interview them or their colleagues, I heard vague references to government work, classified projects, years they couldn't discuss. One particularly interesting case was Dr. Patricia Moreno, who had been a rising star in cellular biology in the late 1940s. She published groundbreaking work on genetic transfer in bacteria in 1948 and 1949, then vanished from academic literature until 1957. When she returned, she was working on agricultural genetics, developing disease-resistant crops. I found her obituary from 1994, and it mentioned that she had worked on classified biological research for the government, but provided no details. I contacted her daughter, who told me her mother had never discussed that period of her life. She had told the family only that she'd worked on important medical research that remained classified, and that she'd signed agreements preventing her from ever speaking about it. The daughter said her mother had seemed troubled by whatever she'd done during those years, sometimes waking from nightmares but refusing to explain them. These patterns suggested a coordinated program, recruiting top biological researchers into classified work during the crucial period following the 1947 recovery. If you wanted to understand what you'd recovered, if you wanted to attempt reverse engineering or genetic analysis with 1940s technology, you'd need the best minds in the field working in secret. By 2008, I had assembled enough information to construct a comprehensive hypothesis about the specimen's origin. I couldn't prove it definitively, but it explained all the evidence I'd encountered. I documented this hypothesis in a private journal, knowing I could never publish it through official channels. The hypothesis was this: a non-human intelligence with advanced biotechnology had been collecting genetic samples from Earth's biosphere for an extended period, possibly decades or centuries. They used this collected material to engineer biological entities specifically designed to operate in Earth's environment. These entities incorporated human and other terrestrial DNA to ensure environmental compatibility, to allow them to survive in Earth's atmosphere and temperature ranges, possibly to interact with Earth biology in specific ways. The purpose of these entities remained unclear. They might be reconnaissance tools, biological drones designed to observe and collect information. They might be communication interfaces created to bridge the gap between human biology and something fundamentally different. They might be failed experiments, attempts to create beings that could survive in both Earth conditions and wherever the creators originated. The crashes and recoveries represented not interstellar accidents, but something more complex. Perhaps deployment failures, the entities dying during transport or shortly after arrival. Perhaps intentional placements that went wrong when the craft malfunctioned. Perhaps even deliberate abandonments, leaving these beings for humans to find and study, a form of disclosure through biological evidence. The human response had been to study them intensively while maintaining absolute secrecy. The security protocols, the compartmentalization, the decades of classified research all served to contain the implications of what had been discovered. Because if this hypothesis was correct, it meant non-human intelligence had been accessing Earth's biosphere extensively enough to collect diverse genetic samples, had technology capable of genetic engineering far beyond human capabilities, and had been operating in Earth's vicinity for an extended period. I also considered an alternative hypothesis that I found even more disturbing. What if the engineering had been done jointly? What if human scientists provided with alien technology and assistance had participated in creating these beings? That would explain why recovery events began in 1947, shortly after human nuclear capability emerged. Perhaps our technological development had triggered contact, and part of that contact involved collaborative biological programs. This alternative explained certain anomalies, It explained why the preservation techniques developed so quickly after the first recovery, suggesting prior knowledge. It explained why genetic analysis capabilities and classified programs advanced so rapidly in the 1950s, possibly benefiting from external technology transfer. It explained the infrastructure built immediately after 1947, as if someone knew there would be biological material requiring extensive study. The implications were profound. If humans had participated in creating these beings, then the crashes and deaths represented failures in a program we were part of. The secrecy protected not just evidence of alien existence, but evidence of human collaboration with non-human intelligence, possibly including experiments that violated ethical boundaries. In 2003, I was contacted through an encrypted email by someone identifying themselves as Dr. Ellen Reeves. She said she had worked on similar biological analysis in the 1970s at a different facility, had reached similar conclusions about the specimen's engineered nature, and wanted to compare findings. I was cautious initially, concerned about security violations, but her detailed knowledge convinced me she was genuine. We began corresponding carefully, never explicitly discussing classified material, but sharing our conclusions and reasoning. Reeves had analyzed specimens recovered in 1972 and 1975. Her genetic analysis, conducted with the technology available at that time, had been less comprehensive than mine, but she'd reached the same conclusion about terrestrial DNA integration. She'd spent the subsequent decades researching the same questions I had. Through Reeves, I was introduced to 3 other scientists who had worked on similar projects across different decades. Dr. James Hartford had been involved in the initial 1947 analysis. Dr. Marcus Chen, no relation to Sarah Chen from my project, had worked on specimens in the 1980s. Dr. Lisa Navarro had conducted cellular analysis in the 1990s. Together, we represented a span of research from 1947 to 2000. 5 scientists who had independently studied these beings and independently concluded they were engineered entities, incorporating Earth biology. We began meeting occasionally, always carefully, always aware that we were technically violating our security oaths by comparing classified information. But we justified it by telling ourselves we were trying to understand the truth, that the compartmentalization had prevented anyone from seeing the complete picture, that after decades of secrecy, we had a right to know what we'd been part of. Hartford was the most valuable source of historical context. He'd been a young researcher in 1947, recruited immediately after the first recovery. He said the military and government response had been chaotic initially, no one knowing how to handle what they'd found. But within months, a structured program had emerged, protocols had been established, facilities had been designated. The speed of that organization suggested either remarkable bureaucratic efficiency or prior planning. Hartford also shared something that hadn't appeared in any reports I'd seen. He said there had been a survivor from the 1947 event, an entity that lived for approximately 3 weeks before dying. During that time, military and scientific personnel had attempted to communicate with it. Hartford hadn't been present for those attempts, but he'd heard from others who were. The entity had shown clear signs of intelligence, had seemed to understand it was dying, had made sounds and gestures that suggested attempted communication. That detail changed my understanding significantly. If one had survived temporarily, it meant they weren't just biological specimens, but potentially sentient beings. The ethical implications of their creation, deployment, and death became more troubling. Someone had engineered intelligent entities, sent them to Earth in craft that crashed, resulting in their deaths. Whether through accident or intention, it suggested a callous disregard for the beings they'd created. Our informal network continued exchanging information through 2010. We built a comprehensive timeline, correlated the genetic findings across different specimens, and decades, identified patterns in the recovery locations and circumstances. We were careful never to document our collaboration in ways that could be traced, meeting in person when possible, using encrypted communications that we deleted immediately after reading. What emerged from our collective analysis was disturbing. The program, whatever it was, had operated continuously for at least 6 decades. The beings showed slight genetic variations over time, suggesting refinement or experimentation with the engineering. The crashes occurred in roughly the same geographic region, suggesting consistent deployment patterns or perhaps limitations in the craft or delivery systems. I'm telling this story now, in 2024, because I'm 71 years old, and the weight of carrying this knowledge alone has become unbearable. The other scientists in our informal network have either passed away or, like me, reached an age where legal consequences seem less threatening than dying with the truth unspoken. Hartford died in 2015. Reeves died in 2019. Chen passed away last year. Navarro is still alive, but she's 68 and in poor health. We've discussed going public together, perhaps publishing our findings jointly to lend credibility through numbers. But the legal risks remain real. We all signed security agreements that have no expiration date. Violation could result in prosecution, imprisonment, certainly the destruction of our professional reputations. Our families could face legal and financial consequences. But more than legal concerns, I've hesitated because I'm uncertain about the implications of disclosure. If the public learned that non-human intelligence has been collecting Earth biology, engineering hybrid entities, and operating in Earth's vicinity for decades, the social disruption could be severe. Religious worldviews would be challenged. Government trust would erode further. The realization that authorities have hidden this truth for 75 years would fuel conspiracy theories and paranoia. Yet I also believe people have a right to know. If we're being studied, if our biosphere is being sampled, if entities are being created using our genetic material, humanity should be informed. The secrecy serves those in power, not the public interest. And if there's collaboration between human and non-human intelligence, citizens of Earth should have a voice in whether that collaboration continues. I've spent the past year writing this account, documenting everything I can remember from my time in that New Mexico facility. Everything I learned from declassified research. Everything our informal network discovered. I've been careful to present only my own conclusions and analysis, avoiding direct quotation of classified documents. I believe this approach might protect me legally while still sharing the essential truth. The question that haunts me most is why. Why would non-human intelligence create beings using Earth biology? Why engineer entities that appear designed for our environment? Why deploy them in ways that result in crashes and recoveries? None of the hypotheses I've developed fully answer these questions. Perhaps they're studying us, using biological entities that can survive in our environment to collect information. Perhaps they're attempting communication, creating beings that bridge the gap between their biology and ours. Perhaps they're conducting long-term observation, monitoring humanity's development over decades. Perhaps it's something else entirely, a purpose so foreign to human thinking that we can't conceive of it. What I know with certainty is that the specimens I analyzed were real. Their hybrid genetics were real. The integration of Earth DNA was real. I know that similar specimens have been recovered over decades, suggesting an ongoing program or presence. I know that the engineering required technology beyond human capability at the time of creation. And I know that governments have hidden this truth for 75 years, I don't expect everyone to believe this account. I have no physical evidence to present, no classified documents to leak, no specimens to display. Everything remains locked in secure facilities, protected by security protocols I don't have the authority to breach. All I have is my testimony, my analysis, my professional expertise as a molecular biologist who spent a year studying something that shouldn't exist. But I'm not alone in this knowledge. Hundreds of people, perhaps thousands, have been involved in these programs over 7 decades. Researchers, military personnel, government officials, all bound by security oaths and threatened with prosecution. Some have gone to their graves with the secret intact. Others have hinted at the truth in vague terms, dropped cryptic comments in interviews, left breadcrumbs for those willing to look. The truth is already leaking out slowly. UFO disclosure has advanced significantly in recent years. Government officials have acknowledged that unidentified aerial phenomena exist and that some display capabilities beyond known human technology. Video footage has been released, showing objects moving in ways that defy conventional physics. The conversation has shifted from whether these phenomena exist to what they represent. But the biological component remains hidden. The public discussion focuses on craft, on propulsion systems, on aerial capabilities. No one talks about the specimens, the beings, the genetic engineering. That's the deeper secret, the revelation with more profound implications. Because craft can be dismissed as advanced technology, human or otherwise, but biological entities incorporating human DNA, that forces us to confront uncomfortable truths about our place in the universe and our relationship with non-human intelligence. I've thought extensively about what I want people to understand from this account. First, that the evidence I studied was rigorous and scientific. This wasn't speculation or conspiracy theory. I applied standard molecular biology techniques to biological material and documented the results systematically. The findings were reviewed by other qualified scientists, though compartmentalization prevented comprehensive peer review. Second, that the implications are complex and not inherently threatening. Yes, it's disturbing to learn that non-human intelligence has been collecting Earth biology and engineering hybrid entities. But the lack of overt hostility over 7 decades suggests either benign intent or at least no immediate threat. Whatever is happening has been happening for a long time, and humanity has continued to develop and thrive despite it. Third, that the secrecy has served political and institutional interests more than public safety. The authorities who maintain these secrets claim they're protecting people from panic or social disruption, but I believe they're primarily protecting themselves from accountability, from questions about why the truth was hidden, from loss of control over the narrative. The world has changed dramatically since 1947. We've developed more sophisticated technology, expanded our presence in space, discovered thousands of exoplanets where life might exist. The idea of non-human intelligence is less shocking now than it would have been in the immediate post-war period. People are ready for this truth, even if governments remain reluctant to provide it. I'm sharing this account knowing it may accomplish little. Without physical evidence, it's just another story, another claim from someone who sees they worked on classified programs. But I hope that by adding my testimony to others, by documenting my expertise and the rigorous analysis I conducted, I contribute to a growing body of evidence that eventually forces official disclosure. If I face legal consequences for speaking out, I'm prepared to accept them. I'm old, enough that imprisonment holds less terror than dying with this knowledge locked inside me. My wife knows I'm writing this account. My daughters know I worked on something classified that troubled me deeply. If I'm prosecuted, at least they'll understand why. To other scientists who have worked on these programs, who carry similar knowledge and struggle with similar obligations, I say this: the security oaths we signed serve institutions, not truth. We have a higher obligation to humanity, to the advancement of knowledge, to ensuring that profound discoveries about our reality aren't monopolized by government agencies. The risk of speaking out is real, but the cost of perpetual silence is higher. The truth I carry is this: we are not alone. We never have been. And the evidence has been hidden in plain sight for 75 years. Non-human intelligence with capabilities far beyond our own has been present in Earth's vicinity, conducting long-term observation or study, creating biological entities using our own genetic material. The crashes we've recovered aren't visitors from distant stars, but something stranger, beings engineered to exist in our environment, possibly to bridge the gap between their creators and us. What happens next depends on whether enough people come forward, whether enough evidence accumulates, whether public pressure eventually forces governments to acknowledge what they've been concealing. I've done what I can by sharing this account. The rest is beyond my control. I'm 71 years old, and I've carried this secret for 34 years. Whatever time I have left, I want to live it, knowing I tried to tell the truth, regardless of the consequences. That's all any of us can do when faced with knowledge that changes everything we thought we understood about reality. The specimens exist, the genetic evidence is real, and the implications are profound. That's the truth I carry, the truth I'm finally sharing, the truth that I hope will eventually become part of our collective understanding of who we are and where we fit in a universe far stranger than we've been allowed to know.